now listening to Conferences on Oncology from Children's Mercy Hospital in Kansas City, Missouri. Today is December 9th, 2024. Our topic is Abbas Chapter 8, Lymphocyte Development and Antigen Receptor Gene Rearrangement. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine Pediatrics and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital in Kansas City, Missouri.
Today is December 9, 2024. I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. My email is included here if you have questions. This is Chapter 8 from textbook of Abbas, Lymphocyte Development and Antigen Receptor Gene Rearrangement. Disclosures here, nothing relevant to the talk today. Learning objectives are to expand our knowledge about overview of lymphocyte development, rearrangement of the receptor genes, then we'll talk about B cell particularly, and then T cell. Couple of questions. Why do immunoglobulin and TCR genes undergo rearrangements in B and T cells and not other cell types? Come back to that. Another one, in both B and T cell development, there is a stage where a pre-antigen receptor is expressed. Which of the following accurately describes an important function of these pre-antigen receptors? All right, so talking about lymphocyte development, we talked actually about, we're kind of going in reverse, right? We talked about T cell activation and signaling. We talked about B cell activation and signaling. And so now we are kind of taking a step back and saying, what happens before all the B and T cells go into circulation, meet their antigen, and give rise to response? During that development, the goal is to just see if they are functional or not. And so during development, there are certain phases or stages that each of these lymphocytes have to go through to make sure that it's a functional lymphocyte. So the first step is, we know that these lymphocytes come from stem cells, so they have commit to becoming a lymphocyte and not any other type of blood cell. The next step is that they proliferate. These stem cells that are committed to be lymphocytes proliferate as progenitors, which are called prolymphocytes, and then they become immature committed cells. Then they have to undergo rearrangement of antigen receptor genes. So that's a major portion of what the lymphocytes in development go through. That phase has certain requirements, and these lymphocytes during their development have to undergo certain screening or selection processes that they have to pass to become mature lymphocytes. So the selected cells have to have a functional antigen receptor protein, and they have to have weak recognition. And then once they are those immature lymphocytes undergo selection process, they become mature lymphocytes, and then they might differentiate into distinct subpopulations. So those are the 5 different stages of lymphocyte development. So let's talk about commitment first. So all stem cells, hematopoietic stem cells, are multipotent, which means they can become different types of cells. And some of these stem cells, when they divide, they become they give rise to more stem cells, but also they give rise to either common lymphoid progenitor or myeloid progenitor. Here we are focused on lymphoid progenitors, and depending on what kind of transcription factors that they come across, they may become pro-B cell, pro-T cell, or IL-C precursor, right? So this process is for the bone marrow, which gives rise to either the alpha beta T cells or follicular B cells, some marginal zone B cells. But there are some lymphoid progenitors instead of bone marrow. During the fetal life, we have these lymphoid cells from fetal liver, and from the liver, these lymphocyte progenitors can become B1 B cells or gamma delta T cells. So what decides what kind of cell it's going to be? It depends on the surface receptors as well as transcription regulators. So if it's Notch1, GATA3, transcription factors that become active in the lymphoid progenitor, that cell will be destined to become an alpha beta T cell. Or if it has EBF, E2A, PAX5, these are the transcription factors that are active. Those cells will become pro-B cells. ID2, it will become an innate lymphoid cell. So these factors will result into expression of proteins that are involved in antigen receptor gene rearrangement, and also whether there is access to those gene loci, right? So in the DNA, there are these certain gene areas that are called locus or loci, and next to it are their promoter regions. So these genes have to be in accessible form for these transcription factors to go in there and be able to start the transcription of those proteins. And when those proteins are expressed, they help in certain functions, particularly in these developing lymphocytes. The important proteins that are expressed are the ones that take part in antigen receptor gene rearrangement. So again, kind of, for commitment, the factors that are responsible are cell surface receptors and the transcription regulators that determine whether these lymphoid progenitors will become TB or NK cells or innate lymphoid cells. So once the commitment has occurred, there is proliferation of the committed progenitor cells. And important signals that help in proliferation come from cytokines. For example, IL-7, which has the common gamma chain. IL-7 is important cytokine that helps in proliferation of the cells. And then the precursors, there is a signal that has to go through once the cytokine helps in, say, cytokine binds to its receptor. In the last chapter, we saw that once the cytokine binds to its receptor, there is downstream signaling, and that has to occur through the cytokine receptors, but also there have to be these pre-antigen receptors on the surface of these committed cells, and there has to be signaling through the pre-antigen receptors, and those two signals are needed for proliferation of cells. Otherwise, those cells developing lymphocytes might die. Epigenetics also plays a role. So kind of going through what I was talking about, excess of the genes in the nucleus, we want to talk about how that occurs. So in the our DNA is actually organized in the form of chromatin in the nucleus of the cells. Chromatin includes the nucleosome, which has the DNA and the histones. It also is in association with non-histone proteins and the RNA. All of this structure comes together to make a tight bond, tight structure, and that tight structure has to open up to make sure that there is access of the genes that are available for transcription. When they are accessible is when the transcription factors can act so that they can start the transcription of certain genes. So depending on that, the chromatin structure can be in euchromatin or heterochromatin phase. And so, which means that when it's available for transcription, there are certain processes that can occur. One, there can be transcription, but there are also some epigenetic mechanisms like methylation or histone modification that can help in regulating the transcription of certain genes. Another process that can occur is is uh, the function of non-coding genes. Non-coding genes make non-coding RNA, and those RNAs, such as microRNA or long non-coding RNA, do not make proteins, but they do take part in regulating the mRNA that make proteins. So they can regulate and silence those mRNAs from making the proteins or transcribing. So it's something to keep in mind, that the proliferation is of these committed progenitor cells takes place in presence of certain signals, or certain signals that are important for proliferation, such as cytokine signals and pre-antigen receptor signals. And then it's regulated by some of these epigenetic factors. So we talked about antigen receptor, right? So, and pre-antigen receptor. So we know that for B and T cells, there can be gene rearrangement that can occur during their development. And the final antigen receptor that's present on the surface of B or T cells is the result of gene rearrangement. So the reason for that is, if you think about the genes that are required to encode various types of receptors. So each B and T cell has unique receptor on its surface, and these antigen receptors are responsible for giving the diversity of antigens that they can bind to. So think about, say, about 10 million clones of T and B lymphocytes that are present, each clone with a unique antigen receptor. But if the T and B cell genes are trying to encode for these receptors to make those proteins that go on the surface and act as receptors, most of the genes will be used up just for the purpose of making receptors, and the function of T and B cells that require other genes may not be. It might not be possible to have those genes. So the alternative idea was, or theory was, that the gene segments in T and B cells can undergo gene rearrangement so that they can give rise to a lot of diversity. And so now we know that that's true. And so something to keep in mind, that this gene rearrangement is occurring before these T and B lymphocytes are completely mature. They come mature lymphocytes are the ones that are going to come in contact with the antigens in the periphery after their development is completed. But then, as we will go through this process of development, we'll see that they might come across some antigens during development that's important for the development of these cells. Regardless, the gene rearrangement occurs before the exposure to the genes. So once that gene rearrangement occurs, there is a selection process. So there are checkpoints where they have these cells have to pass some tests to make sure that they are able to be functional B and T cells before they complete their maturation. So the checkpoints that are present are include one which is called pre-antigen receptor. So when the antigen receptors are formed by gene rearrangement, they undergo gene rearrangement initially in certain genes. For T cells, it's in TCR beta gene locus, and in B cells, it's in the Ig heavy chain locus. So remember, the T cell receptor is made up of alpha chain and beta chain. We are talking about alpha beta T cells. So the initial gene rearrangement occurs in the beta gene locus. Similarly, for B cells, the B cell receptor is made up of IgM. IgM has a heavy chain and a light chain. So the initial rearrangement will occur in Ig heavy chain locus. Once this gene rearrangement occurs, it provides signal for survival, proliferation, and maturation. So let's look at this figure here. So the pro-B, so stem cell was differentiated into the pro-B cell or pro-T cell. Then it underwent proliferation, and then it undergoes that pre-T cell or pre-B cell antigen receptor. Once that pre-B cell receptor or pre-T cell receptor expresses that chain on the surface of the cell, it's not a mature receptor, but it's a pre-cell receptor. It does so in form of single chain, and it associates with a surrogate light chain or surrogate chain, which with that on the surface, these cells will undergo proliferation again, and then these cells have to show that this pre-antigen receptor on its surface is able to send a signal downstream. If that signal goes through, then it provides signal for this cell to survive, proliferate further, and mature. If these cells cannot show that their signaling can go through this pre-antigen receptor, then they will undergo cell death. Once that process, that's a checkpoint. So anywhere there is a checkpoint means that if it cannot pass that test, it's going to die. That cell is going to die. So if the pre-antigen receptor is not formed, it's going to undergo cell death. If it expresses the pre-B cell or pre-T cell receptor but cannot undergo signaling through it, it will again die. And once it can pass through those pass the signaling test, it's going to mature. So these immature B or T cells will express a complete antigen receptor. So now if it's a B cell, it will express heavy chain and a light chain, which will be an IgM molecule, which will be your B cell receptor, not a pre-B cell. Now it's going to be a B cell receptor. For T cells, it's not just going to be a beta chain with surrogate chain, it's going to be a beta chain and alpha chain, which will be our T cell receptor. So that's your, that's going to be expressed on the surface. For T cells, they have to undergo further testing in terms of positive selection, where the T cell meets with the peptide antigen and the MHC molecule on the antigen presenting cell. If it comes across a cell that with MHC class 1, it's going to become CD8 positive T cell. If it comes across MHC class 2, it's going to become a CD4 T cell. So that's the first problem. If it cannot identify or cannot recognize MHC molecule at all, it's going to die. So that's another checkpoint. Similarly, B cells can undergo positive selection, where B cells have to interact with weak recognition. It has to interact with the antigen. So if it cannot recognize it, it's going to die. Or we'll come back to that. For negative selection, so that's a second test, these cells have to be able to bind to that antigen with weak recognition. If it binds too tightly with high affinity, these are going to be self-antigens, so these cells will die because you don't want self-reactive cells to survive and cause problems for the body. So that's called clonal deletion, which means, so that's negative selection. In terms of B cells, there is a process where if it recognizes the antigen too tightly with high affinity, in those cases, those B cells get a chance to modify the receptor on the surface. So go through that gene rearrangement process again, that's called receptor editing. So it will edit its receptor so that it's not going to bind to the self-antigens that with that high affinity, so that they are not self-reactive. If it can undergo that receptor editing successfully, then it will make a receptor that's not as tightly bound, and then it's going to mature. So that's your mature T cell and B cell that are going to be developed at the end of these selection processes. All right, now we go and talk about antigen receptor gene rearrangement. So we talked about commitment of the lymphocyte progenitors, then we talked about proliferation and what signals are needed for that proliferation, and then we talked about pre-antigen receptor, and then we talked about selection processes. All right, so now we move. Any questions at this point? We're going to talk about gene rearrangement after this. Before that, I just wanted to stop and see if I can answer any questions. Okay. All right, moving along. So we know that the gene, we are talking about the DNA, right? We are talking about the genes on, these are not proteins. We are talking, so far we talked about the development process and selection process. We were talking about proteins. Here we are talking about the genes. So in the T cell or the B cell, in the lymphocyte, the DNA has gene segments. These gene segments that make the T cell receptor or B cell receptor have gene segments called variable regions, diversity segment, joining segment, and constant segments. Here we are talking about immunoglobulin locus, so B cells. In those cases, there are 3 of these regions on different chromosomes, immunoglobulin, so IgH or heavy chain locus, and the light chain locus. There are 2 light chain, kappa and lambda, so there are 3 different regions here. So in those areas, there are various V, D, J, and C segments. So multiple V segments, multiple D segments, multiple J segments, and multiple C segments. Similarly, at the immunoglobulin kappa locus, there are going to be multiple of those V, J, and C. So for the light chain, there are no D regions. So for the lambda region as well, there is V, J, and C regions. So as you see, for each of these locus, there are multiple of each. So for variable region, there are about 45. For the heavy chain locus, diversity segments are 23, so on and so forth. So similarly, for the light chain, there are less number, but still those V regions that are V segments that are present. All right, so when you think about the molecule of immunoglobulin, it has the heavy chain and the light chain, and they come together to form the V region and the constant region. So if you think about the structure of immunoglobulin and one heavy chain, and so each immunoglobulin molecule will have 2 heavy chains and 2 light chains. But for this discussion, we are talking about how one heavy chain and one light chain come in contact with each other. So for that, for the light chain, it's going to be the variable region coming in, or segment coming in contact with the J segment and associate itself with one constant chain. In terms of the heavy chain, there is a V region, D region, and J region. So V, D, and J segments that come together and then associate themselves with constant regions, and there are 3 or 4 constant segments attached on the heavy chain. So that's for the B cells. For the TCR receptor, we know there is an alpha chain and a beta chain. And for the alpha chain and beta chain, again, there are going to be the variable, so V segment, D segment, and J segment for the beta chain in contact with that constant chain. For the alpha chain, it's similar to the light chain, so there is no D region, but there is alpha variable and J segment along with the constant segment that come together. And so then these chains fold over each other to give that tertiary structure of TCR or immunoglobulin molecule. This is just going over the same thing for TCR, alpha, and beta, multiple VDJ and constant regions that are possibilities for them to make the final segments. And then for gamma delta, similarly, there are all these regions for each of these molecules. So if alpha and beta are going to make segments are going to undergo gene rearrangement and make a successful gene rearrangement, they'll give rise to alpha beta TCR alpha beta T cell, versus if the gamma delta undergo the rearrangement. So as you see, there is less variability in gamma and delta chains. If that's the case, they'll make the gamma delta T cells. So when you compare all the other cells versus T and B cells, we know that the T and B cells undergo gene rearrangement and not all cells do that. And we'll see that the reason for that in a minute. But in general, there is one variable segment, one D segment and one J segment that come together to form from the germline DNA in the T cell. This is the germline DNA, which will have all those variable segments, D segments, J segments, and constant segments. They'll undergo rearrangement so that there is one V segment, one D segment, and one J segment that come close to each other along with the junctional diversity basis, and then they come in contact with that constant region. That rearranged DNA will undergo transcription to form the mRNA, and then give rise to the protein, which will have one variable D and J segment to make that cell receptor. So we know that there's for the light chains, the rearrangement occurs initially in the V to J segments, versus in heavy chains, the first rearrangement will occur so that there is binding of D to J segment. Once that happens, the variable region will then undergo gene rearrangement so that V is bound to the D and J segments. So DJ and then VDJ. All right, talking about how this recombination occurs. Remember, these are genes. They can be in the wound up position where their genes are not available for transcription, versus then they can open up so that the genes are available for transcription. When that happens, there are two helices of DNA, right? We know that there are two copies of each gene. Those are wound up, so the chromatin has to open up in the region of gene rearrangement, and two specific segments come close to each other. Once they come close to each other, there are double-stranded breaks that occur on both the sides, both the strands, and then nucleotides are added. Some of them are removed at those broken ends. Once that process occurs, both the sides are ligated, they are sewn together, right? And then the final DNA that makes the mRNA becomes apparent, that will undergo transcription. So then there is transcription of the leader sequence, so VDJ exon, then there is that intron in between J segment and constant region, and then there is the C region that is transcribed. That's your primary transcript. Once that primary transcript has occurred, there is splicing of the RNA to form the final RNA transcription that's going to make the protein that gets translated on ribosomes to produce the protein. In this case, the protein is the antigen receptor chain. So it will make the beta chain in T cells, or the heavy chain in terms of the B cells, that is going to be produced at the end of this VDJ recombination. So again, we started with the germline DNA in the T cell or B cell that had multiple V segments, D segments, J segments. That undergoes recombination so that there is a break that occurs so that one of these variable region will come in contact with, so initially D and J, one of these D regions will come in contact with the J region, and then those two, D and J, will have one variable segment come in contact with them so that they'll form that primary transcript along with the constant region. So how does the DNA know where to make those breaks? Where does the rearrangement occur? And so there is a very specific process that occurs in this DNA. There are recognition signals for this recombination to occur. And those signals are called recombination signal sequences, or RSSs. These are unique to just the immunoglobulin genes or TCR genes. So clearly not all genes undergo recombination, and the reason is they don't have these RSS. Recombination signal sequences have very specific structure. They include a heptamer that has certain bases. You see the structure here. That's called a heptamer because it has 7 bases. And then there is a spacer. There is a 12 base pair spacer or a 23 base pair spacer, as here. Those spacers are between the heptamer and nonamer. Nonamer has 9 bases, so it's called nonamer. Heptamer has 7 bases, so it's called heptamer. And in between those are the spacers. Those spacers can be 23 base pairs long or 12 base pairs long. So when the double-stranded break occurs in these DNA strands, it occurs between a heptamer and a coding sequence. So if you look at this DNA, there is a 5' and 3' ends of it. Here, when you see that heptamer, on the left of that is the variable region. So that's the coding segment. Or on this side here, there is a heptamer, and on the right side of that, there is a J segment. So, or there is a D segment here between the heptamers. So the double-stranded break is always going to occur between the heptamer and the coding segment. And then once that break occurs in between the heptamer and the coding sequence can bind to each other. So here, say, the D and the J segment might come close to each other and form that transcript, versus the rest of the RSS, recombination signal sequence, which has the heptamer, the spacer, the nonamer, all of that will make a circle and will be removed. So that's one type of process that can occur for the recombination. The other type is where sometimes the way the DNA is rearranged, for example, between V and J segments, they are facing, the nonomers and heptamers are not facing each other, they are facing in the same direction. In those cases, there again, the break always occurs between the heptamer and the coding sequence. So when that happens, there is inversion of the DNA helix, so that those two coding sequences can come close to each other and undergo recombination, and then the rest of that RSS will be attached to the end of it. So either of those can occur, inversion or formation of a circle, but regardless of that, the double-stranded break is going to occur between the heptamer and the coding sequence. This is just showing the promoter and enhancer region. So say there is a V and J combination that occurs, and there is recombined DNA. When that happens, there is a promoter region and enhancer region that are going to come close to each other just because of the process of recombination, because that intervening segments are going to go away. So because those intervening segments are going to go away, let's see, I'm looking at this here, say the V region, V1 region or V2 region is going to come in close contact with the D or J region and then the C region, and the intervening segments are going to go away. So the promoter and enhancer are going to come close to each other. That's going to help with transcription. All right, questions at this. This was all very basic information about that VDJ recombination, of how the RSS is important part of that VDJ recombination. But now we're going to talk about specific steps of VDJ recombination. Questions? All right. So here you go. You see all these things in red that you guys know that are important terms to memorize. Let's go back to our 5' and 3' DNA segment that has the heptamer, nonamer, and it has, say, V and J segments. All right. So we are talking about V and J segments. The steps of recombination include the first step being synapses, which means the antigen receptor genes are made accessible to the proteins that are important for the recombination process. So for that, there is a formation of loop of chromosome to bring together the selected DNA segments or that have to undergo recombination. So in this case, say V and J, right? So V and J come close to each other, and to do that, this part of the DNA in between them is going to make a loop. So these two come close to each other, which is called synapses. At that edge of synapses are proteins that come close to each other for making double-stranded breaks. They are called RAG1 and RAG2. RAG1 and RAG2 are also together called VDJ recombinase. So RAG1 is the active enzyme that makes the break. You think of the scissors, and RAG1 and RAG2 are the scissors that make that break in the DNA. RAG1 has that activity of the break, making that break, and RAG2 is the regulatory part that's needed for the function of RAG1. So defect in RAG1 or RAG2 can lead to immunodeficiency, where these cells cannot undergo recombination. So RAG1 and RAG2 recognize the junction between the heptamer and the coding segment and make that break. So when that happens, these two segments will come close to each other, and that break is going to occur there, and that will form this hairpin structure that forms a covalent hairpin and holds these two close to each other. That's the function of RAG1 and RAG2. Now we want to bind these two segments, and to do that, these hairpins have to open up. So for that, there is another enzyme that's needed at these hairpin ends that opens up the hairpins, called Artemis. That Artemis will open up these hairpins, and then there's another enzyme that comes along called TDT, terminal deoxynucleotide transferase, that will add some bases to these ends. So that's the function of the TDT. Once that's done, the next step is joining these ends to each other to complete the process of recombination. This joining occurs because there are enzymes and proteins that take part in that DNA repair. So those proteins include, and enzymes include DNA PK, KU proteins called KU70 and KU80, and the DNA kinase will phosphorylate. It's a kinase, so it adds a phosphate molecule. We saw that last week. So phosphate molecule is added, so it phosphorylates and activates the Artemis that's involved in end processing. That's the enzyme of DNA PK enzyme. And then there are these other enzymes that help in pretty much sewing up that area or repairing that area. So these are DNA repair genes. The ligation is done by DNA ligase 4 and XRCC4. So DNA repair genes are needed. All these proteins are needed for that last process of joining. So what did we see? Recombination occurs where the segments come close to each other, which is called synapses. At those nonamer and recombining segments edge are the proteins or enzymes called RAG1 and RAG2 that makes a break. Then the Artemis comes along at that hairpin end and opens that hairpin up so that the TDT can add some bases for giving diversity. Once the TDT does its job, there is going to be joining of these two segments using all these proteins and enzymes, including DNA PK, DNA ligase 4, XRCC4, including Ku70, Ku80 proteins. So Ku70, Ku80 just binds to the end of the breaks and then brings in, they are like adapter proteins that bring in the DNA PK that is the repair enzyme. And then once it does its job, the DNA ligase 4 and XRCC4 come along. So the ends are ligated and it's not homologous, which means both the ends are not going to be exactly the same. That's called non-homologous end joining, or NHEJ. All right, something to memorize. I think this figure does a great job. So if you want to kind of look at this figure, pay attention to all the details in the figure, it will make it easier to memorize those steps. All right. So talking about diversity, the whole point of making sure that we use this recombination in B and T cells is to provide the diversity of B and T cells. The diversity of B and T cells comes because of the diversity in their receptors. So these receptors that are made from VDJ recombination have various parts of their structures that are diverse compared to the others, right? That's how the diversity occurs. So there are different types of diversity that you have to keep in mind. So there is that combinatorial diversity, which means a random V region is going to come in contact with random J region. There's going to be a random D region in terms of the heavy chain or the alpha beta, or sorry, the beta chain. And so these random segments, it could be V1 for one of them, it could be V7 for one of them. So any of these random V segment is going to bind with a random D segment and a random, which one did I leave out? J segment, to give that combinatorial diversity. The junctional diversity is also provided. Remember, we talked about that addition of nucleotides at those ends by TDT. There are nucleotides that are added to the junction of those segments that provides the junctional diversity. So there are two types of segments that are, or nucleotides that are added to the ends by TDT. One is called based on the template. So we'll see that in a minute here, how that's done. But they are called P nucleotides if there is a template present, and N nucleotides that do not have template. The N nucleotides are more in the heavy chain and the beta chain or the gamma chain compared to the light chain or the alpha chain or the delta chain. So that's your junctional diversity. So we'll look at this more in detail here. So combinatorial diversity, I think that's pretty clear. You just combine any random segment to another to give that variability. The junctional diversity is by the N and P nucleotides. So let's look at that information. So here is the double-stranded DNA. The break is made and these are the hairpins that are created. This is a closer view, a zoomed in version of what we saw in that VDJ recombination processes. So when that hairpin is formed, it occurs that there are these two segments that have all these bases that make the DNA structure, right? So here, say the hairpin opens up. When the hairpin opens up, it doesn't do a good job of making that break at the same place. So Artemis opens up the hairpin, but it's not perfect. So these are uneven ends. When that uneven end occurs, it gives a template to which more bases are added. So if those bases are added based on this template, which means for A, there's going to be a T added, for a C, there's going to be a G added. For a T, there's going to be A added, and for G, there is a C added, right? So based on the template, if the nucleotides are added, to these uneven ends, they are called P nucleotides. And then in between these two ends, there is still some space, and we need to stitch that up, right? We need something in between them so that it's continuous. And those nucleotides that are added randomly are called N nucleotides. So these are the P and N nucleotides that give the highest diversity to the chains. And that makes the CDR3 region. Remember, that's the most hypervariable region. Among all the hypervariable regions, CDR1, CDR2, CDR3, CDR3 is the most variable. And so this process is what gives that diversity to that region. That also is used clinically in clonality determination. All right, questions about that? Now we're going to discuss more cell-specific information. So remember, the processes that we just talked about occur in both B and T cells. But now we want to put that into context of the cell development. These processes of VDJ recombination occur in the B cell or they occur in the T cell, and their job is to make the TCR or the BCR. But the overall development of the B cell has to go on beyond that development of the receptor. So we want to see what that looks like now that we know that that recombination is occurring during that development process. So let's talk about B cells. So as you see, there are various stages of maturation of B cell. Initially called a stem cell that becomes a progenitor of B cells, pro-B cell. That becomes a large pre-B cell and then a small pre-B cell. Then it becomes an immature B cell and then finally becomes a mature B cell. So there are various stages of these maturation. Lot of these processes occur in the bone marrow. However, the immature B cells will be released into the periphery from the bone marrow, and the B cells finish their maturation in the periphery. During this time, remember, there are these proteins that are going to be expressed that will help with VDJ recombination. They'll make that pre-BCR, and then they'll make the BCR, the mature BCR. And each of these stages of maturation, there are certain proteins or molecules that are expressed in these stages that will define the stage of maturation. Which means when that VDJ recombination is going on during pro-B cell and large pre-B cell stages, you'll see a lot of RAG expression and TDT expression. But once that process is done and the BCR is made, the RAG and TDT are no longer required. So these proteins are no longer going to be expressed in a mature B cell. So if you find a cell and you do its studies to see what it expresses and you find RAG and TDT in them, they're likely going to be the pro-B cell or the pre-B cell stages of B cell compared to a B cell that's more mature when there is no RAG or TDT in it. Similarly, the various immunoglobulins that are expressed, the pre-B cell, it expresses the Ig heavy chain along with the surrogate light chain, and then it expresses mu heavy chain inside the cell, so it's cytoplasmic, and then that IgM goes to the surface to make the IgM on the surface. That's your immature B cell. And when there is IgM and IgD, both are present on the surface, that's a mature B cell. So each stage is recognized by what is expressed on those cells, and the cell surface markers are mentioned here as well. All right, similarly, we want to kind of go over the same process for what we talked about, the recombination. It's initially the DJ recombination, then the VDJ recombination. Once that occurs, it binds to the C region. Mu region is bound to this VDJ segment, and once that transcription occurs, IgM is formed. Once that happens, the C region can be spliced to form whichever chain is needed. So here we are talking about IgM, where there is splicing so that C mu region is attached to it to make the IgM. heavy chain. Pre-B cell receptor, the mu heavy chain, is bound to surrogate light chain. There are different types of surrogate light chains that come in contact with the heavy chain, lambda phi, V, pre-B proteins that make the surrogate light chains. They are bound to the Ig alpha and Ig beta signaling molecules. Once these cells express pre-B cell on the surface, they could undergo proliferation and signaling. If there is downstream signaling, remember we talked about B cell signaling, this is not BCR signaling, this is pre-BCR signaling. This is a developing B cell, pre-B cell stage. If it has a heavy chain along with a surrogate light chain along with Ig alpha and beta, it has to bind to an antigen expressed in that area, and then once the signaling goes through, it has to go through the signaling through BTK. Once that goes through BTK, it gives the signal that this pre-BCR is able to function. Once that happens, it can undergo proliferation and this pre-B cell is going to survive. If this signaling doesn't go through pre-BCR, then this signaling did not go through this pre-BCR, then that BCR has to undergo further gene rearrangement. Once the IgH locus on one chromosome undergoes gene rearrangement, the other copy of the other chromosome has another copy of IgH that is excluded. It does not undergo gene rearrangement. But if this is not successful, the alternate chain starts going through the gene rearrangement. If there is no successful gene rearrangement, then that cell will undergo death. Once the heavy chain undergoes its VDJ recombination, the light chain, kappa chain, will undergo gene rearrangement, so V and J gene segment rearrangement. Once that happens, if the kappa light chain is successful, it will make its light chain. If it shuts off the surrogate light chain transcription at that point, and then so the pre-BCR is then going to end up becoming a BCR. Similarly for TCR, it's a similar process. All right, immature B cells. So kappa light chain gene rearrangement occurs. If it does not occur, the lambda light chain gene rearrangement will happen. That will complete the IgM. It will have the heavy chain and the kappa chain. If not, it will have the lambda chain. So light chain and heavy chain together will form the IgM and it will be expressed on the surface of the cell to end up from becoming pre-B cell to becoming a immature B cell. When that happens, IgD is expressed on its surface in the periphery so that it becomes a mature B cell. Here there is light chain isotype exclusion, which means either the kappa or the lambda chain will undergo gene rearrangement. And then BCR gives independent tonic signals for survival, so that if it recognizes self-antigen with high avidity, that will undergo receptor editing. It will edit the receptor so it's not self-reactive. If it does not if it's still self-reactive, then it will die. If not, it will leave the bone marrow and then mature in the periphery in the spleen. There are two types of mature B cells, B1 and B2 B cells. B1 B cells are, so there are also some transitional phases. So if it's a bone marrow B cell, it's called B2 B cell. And if it's a liver B cell, it's called B1 B cell. All right, and then from the bone marrow, these cells become the follicular B cells, which are our regular B cells that we talk about. And then there are some marginal zone B cells that end up staying in the spleen. Follicular B cells have IgM and IgD on them. They reside in the follicles and get survival signals from the B cell survival factors such as BLIS or BAF. And the B1 B cells we talked about come from fetal liver. They mainly produce IgM. They have limited diversity. Similarly, marginal cell B cells come from bone marrow, but they also are short-lived and IgM producing cells. Positive selection. We talked about this, that the B cells express Ig molecule on the surface. If they have high avidity for self-antigen, they'll go undergo receptor editing, and if not, they'll undergo death. We talked about that. All right, so that's B cells. Let's quickly talk about T cells. T cells undergo development in the from after bone marrow in the thymus. So similar process for T cells as we talked about for B cells. Each of these stages, the expression of certain proteins and enzymes is specific, and surface markers are specific for each of these stages. From the bone marrow, they end up in the thymus. That's where they undergo the maturation process. And then the mature T cells, single positive cells, are going to be released in the periphery. Thymus has thymocytes. These are the developing T cells. They move from the cortex, so outer region, to the medulla of the thymus. The cortex has thymic cortical epithelial cells that help in the development of the thymocytes, and in the medulla there are thymic medullary epithelial cells, or TMECs, that help in the development of these T cells. There are also some dendritic cells, macrophages, that can have MHC class 1, class 2 on them, and they help in the development of T cells. So, as they move through the thymic cortex, the thymocytes express CCR9 that are attracted to the thymic cortex that have CCL25. As they express CCR7, they are moved towards the gradient of CCL21 and CCL19. So, and then once they are mature, they actually express a CYPR receptor, and that helps them to leave the medulla and enter the bloodstream that has CYP in it. So the cell with that receptor is attracted towards that gradient. That's the movement of the T cells. So same thing, during their maturation, they undergo the DJ and then VDJ recombination. 90% of the double negative T cells give rise to gamma delta T cells, 10% give rise to, alpha beta T cells, and 10% give rise to gamma delta T cells. Unlike the B cells, where the C gene switches from C mu chain to G, A, or E, giving rise to IgA, IgM, IgG, IgE, that C gene in the T cell does not switch. We talked about the stages of T cell. So the pre-TCR is similar. The TCR beta chain is expressed on the surface with the surrogate chains like pre-T alpha chain. CD3 and zeta chains are their signaling molecules. The signal has to go through those signaling chains in this pre-TCR stage so that it survives and proliferates. If it does, then it will go on to become a double positive T cell, which will have it will express the alpha beta TCR and delete the delta locus on that, and it will start expressing CD4 and CD8 on their surface. Once it undergoes, it comes in contact with MHC class 1 or class 2, it will become CD8 or CD4 single positive cell. So it enters the medulla, and as you see, these double positive T cells eventually are going to start becoming single positive cells. That selection process occurs in the medulla. So initially, the positive selection occurs where it comes in contact with one of the MHC. That's called MHC restriction, which means if it's MHC1, it's going to be CD8. If it's MHC2, it's going to become CD4. So there are certain transcription factors or regulatory factors that play a role for that, such as RUNX3 activates expression of molecules that are important for CD8 T cell compared to CD4 T cells for other cells. And then the second process occurs in the medulla, where it undergoes negative selection, which means that it has to bind to the cells with self-antigens with light avidity, but if it binds too tightly, then those cells, T cells, developing T cells, are going to undergo apoptosis. Thus, thymic medullary epithelial cells and dendritic cells and macrophages express a gene or a protein called AIR, and AIR helps in presenting self-antigens, tissue-specific antigens, to these developing T cells. If AIR is defective, you can have a lot of autoimmune problems. This is called autoimmune regulator, AIRE, and autoimmune regulator, if it does not do a good job, if it's defective, then it can give rise to autoimmune disorder called APISET. All right, once the positive and negative selection is successful, it can help in giving rise to single positive T cells. Last couple of slides here, T cells, we talked about the alpha beta T cells. There are also gamma delta T cells with limited diversity, and initially the gamma chain undergoes rearrangement, and then the delta chain will do the same. These are about 10% of the T cells. There are also another type of cell called NKT cells that have that develop in the thymus. All right. Questions? Why do immunoglobulin and TCR genes only undergo rearrangement in B and T cells, not in other cells? Anyone?
Is this C because of RAG1 and RAG2?
Yep. RAG1 and RAG2 are expressed in developing lymphocytes. In both B and T cell development, there is a stage when a pre-BCR or TCR is expressed. What describes the important function of pre-antigen receptors? What's the function of pre-BCR or pre-TCR?
I think this one is A, positively select for lymphocytes.
That's correct. So it helps in positively selecting for lymphocytes that have successfully undergone a first round of VDJ recombination required to express functional antigen receptors. All right.